It's my pleasure to welcome all of you to this PILS lecture with a focus on how to model microbial dynamics and metacycling. And it's my pleasure to give a glimpse into how we do this modeling. And so what I think modeling can provide is that, so why is it important to do soil systems modeling? When we look at the ecosystem services that are provided by soils, such as climate regulation or the buffering and filtering services that are provided by certain soil functions, all that is driven by underlying biological, physical, and geochemical processes that you can see here at the bottom. And it's important to understand these processes and to quantify them and to predict them. And here, soil systems modeling can really help. And in my focus today, I will have a slight focus on the biological processes, but also touch on physical and geochemical processes because all of that is intermingled. So when we now zoom in a bit on the biological driven processes in soil, we can see that the soil is a habitat for soil organisms, and this controls certain process clusters that are biologically driven that you can see here, and they are, so I will not read all of them, but they are related to carbon storage. And I want to say here also to plant disease control, so that refers to root-soil interactions here already. Then one big process class is also biodegradation of contaminants, which is related to the water storage and purification function of soils. And of course, also there are process clusters that are related to nutrient cycling, and all of that affects at the end also plant productivity in agriculture, agroecological systems. So in general, you can distinguish between mechanistic and data-driven modeling approaches as two extreme ends. And the big advantage of mechanistic modeling approaches is that this enables to formalize postulated mechanisms in the model and allows to reflect causal relationships and the knowledge on processes in soils. Whereas on the other side, these data-driven approaches that are used a lot in PhenoRob as well, they are used for interpreting data and recognize patterns, but also for many others. So, and in reality, the soil systems model that we work with are not fully on the mechanistic side because we are simply lacking the knowledge to be able to set up such models. They are somewhere in between the mechanistic and data-driven, but more on the mechanistic side. And what you can see here is a typical iterative model development procedure, or an idealized model development procedure. And it starts with modeling conceptual in developing a conceptual model and then implement it into a numerical model. And these models can be either have no spatial dimension, as you can see here, when you consider a homogeneously mixed system. You can account for spatial dimensions either in 2D or up to 3D. And you can easily see that the models then already become much more complex because you need a lot of information, and also you need the process descriptions to describe these all these processes in 3D. Then inside the model, you consider for certain biogeochemical reaction pathways. This is shown here on the example of a nitrate denitrification. But this relates also, of course, to all kinds of turnover of organic substances. Then the models can be developed with different spatial and temporal scale in mind. So to have typically models that consider only small spatial scales have also small time scales, because processes that are relevant at small spatial scales are typically also very fast. And then after this, you have a numerical model, and that is implemented in a programming code as well. Then you need to define the parameters of the model, or you can either measure them or infer them by model calibration. That is this part here. And I will not go in all the details. You need, of course, mathematical algorithms for this model calibration, for example. And the outcome of the calibrated model is that you test the model against data to decide if the model can convincingly reproduce or explain the observation data sets. And if that is not the case, you have to go back to the model development step, and you end up in an iterative cycle here. If you can reproduce the behavior, your work is not done yet, because you also have to look how well the parameters of the model are identifiable and what are the critical controls of the simulated processes. And that's this field of related to parameter identifiability. And from that, you can really gain information on where the knowledge on the system functioning is currently limited. And also from there, you can go back to generating new experiments and also developing new models. So in my talk today, I will focus on that first part here, how we design models. And I will also not go deeply into the numerical implementation. So if you look at soils and how they are embedded in nature, you can see that soil is a multi-scale porous media. So there are processes taking place where the relevant scale is at the pore scale here. And this affects processes up to the landscape scale. So, and that means processes at different scales are intermingled. And you can see from that already that it will be quite difficult to describe all these details that happen here at the pore scale up to the landscape scale. And therefore, soil systems modeling is also always related to finding mathematical representation of these upscaled mathematical representation of these pore scale processes that are still handleable in our models. And what we typically deal with are models at the continuum scale. So we aggregate effects at the single scale, microbial cell scale, and pore scale to the continuum scale, which you can see here. And yeah, with a view on microorganisms, so we come from these individual microbial cell scale, where you have a typical interaction distance between microbial communities of about 20 micrometers, to a microhabitat scale, which is also relevant for processes at larger scales. So in this case, it's, for example, the rhizosphere that has a dimension of about in the range of millimeters. And this is then aggregated to the core scale and even larger, as you can see on the left side. Okay, and to describe processes at the continuum scale, I put the mass of soil at the continuum scale here on one slide because we have essentially three master equations to describe water flow, which you can see here. With the so-called Richards or Richardson equation, because this equation was actually first published by a researcher called Richardson, and then 1 year later by Richards, but it's mostly known as the Richards equation. For details how to derive that equation, I can recommend the PILS lecture by Stefan Collett. Then we have a second master equation that describes heat flow in soil. And a third one that describes the reactive transport of chemicals. That's the convection dispersion reaction equation. And the basic structure in all these equations is that you have a transport term, which is this one here, and a reaction term plus a reaction term, which is a source sink term, here either for water, for heat, or for chemicals. And it's via these source sink terms are controlled by microorganisms and plants. And for the plant control, I can recommend the PILS lecture on functional structural modeling of plants by Andreas Schnepp. So although these equations look quite easy, there's a lot of complexity hidden behind the terms of these equations. And in this lecture, I will focus on the reaction term, mostly on that reaction term of the reactive transport equation here. So when you look at reactive transport, there are several processes that are part of this transport term here. That's diffusion. So the main processes here are diffusion, convection, and dispersion. And the main process here in the that defines the reaction term is the biogeochemical transformation of organic and inorganic chemicals. And this reaction term in general is a function of time, space, and the soil profile of the concentration of chemicals in the solution phase. If we make the assumption that microbes can only access chemicals that are dissolved in the liquid phase in soil, then it's so this reaction term is also dependent on the microbial abundance, and it is dependent on a number of parameters that we have to either measure or identify using inversion techniques. So we can include the control of biogeochemical reactions on the microbial abundance and microbial dynamics at different scales again. So we can even do that at the Earth system scale, and it has been shown that considering for different functional types of microbes, so these are selected microbes that are shown here in that example model, and the K-selected microbes differ in their growth rates and their use of soil organic matter, that this can improve predictions of climate change effects on soil organic matter storage and on CO2 release. Then at the ecosystem scale, we can have models that consider for trophic networks. So you see here also degradation of soil organic matter that is used by fungi and bacteria, and here comes a trophic network in, and with that you can also consider for the top-down control of trophic networks on these mostly microbially driven biogeochemical reactions here. And at the community scale, you can consider, that's the scale where you have models that consider for soil-plant interactions, also with different functional microbial types, as you can see here. And then you can even there are even models that go down to the pore scale, where you really consider for interactions between individual organisms. So for example, between fungal hyphae and bacteria, and with the solid soil constituents. If you now take a bit broader view again on a soil profile, which you can see here on the right, we can see that with respect to soil organic matter turnover, so I can really recommend, if you want to get an overview about modeling approaches that are used in to simulate soil organic matter turnover, I can highly recommend to look into that really comprehensive review on existing model approaches by Stefano Manzoni and et al. Although that is quite some years old, it's still relevant, and the modeling approaches that are in that review are still used today. So in general, so what you see here is a conceptual scheme, how soil organic matter turnover is reflected in soil organic matter models. So you have the models reflect different organic substrates, and that can be either done as a continuum representations, but most models differentiate the soil organic matter pools into different individual compartments that have, for example, a certain CN ratio and that have a certain degradability. The input to these organic substrate compartments is simulated using input flows by litter fall, root decay, and exudates, which is the main input pathways of soil organic matter into soil. Then the soil organic matter is assimilated by the microbial community and also by the faunal community. And here again, you have mostly this compartmental approach distinguishing different microbial pools along the soil profile. The microbes can decay and become part of and this is an important process for soil organic matter formation. Then microorganisms can mineralize organic compounds to produce, for example, ammonium and nitrate, which can be then taken up by plants as a nutrient. And of course, you can have also additional inputs by land management due to fertilization, for example. Microbes also immobilize nutrients, as shown here for the example of nitrogen, by incorporating it into their biomass. And with that, you get soil organic matter cycling of that is largely microbially driven. So we have seen a great advance in molecular and genomic techniques that provide a lot of data on the regulation of biogeochemical pathways, starting from the genome of individual microbes up to the proteins that can be produced based on the genes that are within due to from the genes that are contained in the genome. And we have also measurement techniques that inform us on how this microbial genome translates into functions and processes at the community scale and ecosystem scale. So while this review here is already a few years old, so more than 10 years old, it's still a challenge to really use that data with modeling and to make it usable for process-based model predictions. And that's something where my group works in. We want to improve the use of such data to make better predictions. And I will show you some examples now from my research. And the first example that I will show you from a bit older work already is that how we can use models for soil organic matter and coupled soil organic matter and pesticide turnover. And here we concentrate on understanding pesticide degradation in the detritosphere. So the detritosphere is a microbial habitat which comprises the soil-litter interface and the adjacent soil. And as I said before, that's one of the interfaces where we have a major input of plant-derived carbon into soil. And we performed an experiment here, a lab experiment, where we brought the detritosphere in the lab using such soil cores of 3 centimeter height, which had a litter layer on top, as you can see in that image here. And we wanted to understand if the additional supply with resources and energy from the litter carbon affects the degradation of a pesticide, MCPA, where you can see a structure. So that's a broadleaf pesticide that is also widely used in agriculture and still used also. And we wanted to understand the effect of the additional carbon supply on 2 degradation mechanisms. One is that specific pesticide degraders use this compound as a growth substrate, or the other would be a more unspecific degradation mechanism, which is the co-metabolic transformation of MCPA. And we had some evidence that this additional supply with resources and energy can accelerate the MCPA degradation, which I will show you also in a second. So to interpret the data and better understand the system, we set up soil organic matter, a coupled soil organic matter and pesticide turnover model, which accounts for dissolved organic carbon, what you can see here on the right, in 2 different pools with different degradability, high quality dissolved organic and low quality dissolved organic carbon. We account for sorption of these pools, and these 2 pools are fed from the litter input due to transport from the top into the different soil layers here. Then we reflected in the model a quite relatively complex structure of the microbial biomass. We distinguish between fungi, bacteria, and pesticide degraders, and we account also for the activity of microbes, because not all microbes are typically active in the soil, but the part of the microbial community is dormant. Yeah, then we also had an insoluble soil organic matter pool, which are the non-dissolved compounds, which are also decomposed, and all these decomposition reactions are driven by the abundance of the microbial biomass here. We had, of course, also a pesticide pool, and we also consider here for sorption, which leads to an reduced bioaccessibility of the pesticide carbon to the microbes. And of course, that was a 1D spatially resolved model. We also accounted for transport of litter carbon and pesticides along the vertical direction. Yeah, the pool structure was defined in a way that we can match the model with measured data on the abundance of fungi, bacteria, and also specific degraders, and we used certain functional gene markers for that. And here you can see some results of the model for the 13C, the relative 13C enrichment in the total organic carbon. That's what you see in the first row. So that's more or less the litter-derived 13C in soil organic matter in different soil depths. And you can see that the model can simulate that the measured dynamics quite well. So the orange one is a treatment where no litter was placed on the top of the soil core, and you see the effect of the litter addition here clearly, and the model can track that quite well. We can also simulate, of course, the dissolved organic carbon in the different layers at certain time points and simulate that quite well. And what we can see if we look at the pesticide, residual pesticide concentration in the soil, we can see that with litter addition, we have an accelerated degradation of the pesticide, especially close to the litter layer, and this diminishes in that direction. If you get lower down the soil core, that means if you have a lower input of dissolved organic carbon from the litter. And we could also measure the incorporation of pesticide-derived carbon, which is this 14C carbon, into the microbial biomass and simulate it. So here you can see that the model has in some parameterization, some an underestimation of the microbial-derived pesticide carbon, but overall the predicted dynamics matches quite well with what we have measured. So I will not go into the details of all these results here, but in principle, the model could also predict the measured abundances of fungi, bacteria, and specific degraders. So that's the specific degraders, that's the bacterial gene marker, and that's the fungal gene marker. With the fungi, there are reasons why the model underestimates the abundance here, and especially in the first few layers. But what you can clearly see is that due to that litter input, we have a strong stimulation of fungal growth. And you can use then the model to get insights into the system to quantify, to estimate quantities that cannot be directly measured. And that's, for example, the MCPA-derived microbial biomass in the total soil core, or how much MCPA was used as a growth substrate directly by specific degraders or indirectly after MCPA had been transformed to fungal co-metabolism. And with that, what we have seen here is that the accelerated MCPA degradation was predominantly regulated by the fungal dynamics and by the co-metabolic transformation pathway, whereas the specific degraders then took up MCPA-derived carbon after the molecule had been co-metabolically transformed by the fungi. And such insight would have not been possible without having such a model. So we went one step forward in a follow-up work where we looked at the gene regulation, what's the effect of the gene regulation on pesticide degradation. And for that, we came up with a model that really also can simulate the transcription from the gene to this messenger RNA to the enzyme and the production of the enzyme. And we have compared that against a number of more simpler modeling variants. And what you see here with the black, so what you see here is also the results of an incubation experiment, which was where we fitted different model variants. And the most complex model variant is this V0 variant, which fits the data best and can also track these gene transcript dynamics as well as the gene abundance dynamics. And with certain simplifications on the, so these more simpler models tested certain simplifications on how complex the representation of the transcript dynamics has to be. And you can see that only the V3 model was more or less okay, but otherwise we really had to consider the complex dynamics to be able to match the measured data relatively well. And also this model gave further insights into mechanisms, in this case here on the relationship between the transcript copy numbers. Maybe concentrate on that figure here for the model V0. So that's the relationship between transcript abundance and the rate of mineralization. So at the point when we did that work, there was often a linear relationship postulated between the transcript copy number and the rate of mineralization, and we could show both in the data but also in the model that we have an historetic relationship. So that means it's really important to know where you are on that curve here. And what you can also see that some of the other model variants, especially these model variants down here, they could not match this historetic relationship, and that means that these are the mechanisms represented in these models are not the right representation of the processes that we need to make reliable predictions. Okay, we can also use models to get insights into pesticide persistence. In that case, we used a model to simulate the degradation of glyphosate, which is the most widely used pesticide worldwide. And to understand a bit better why especially the first metabolite of glyphosate or the main metabolite of glyphosate degradation persists in soil, AMPA. And what you can see here from the experiment with an agricultural soil degradation experiment is that the glyphosate is degraded actually quite fast, but then we have the formation of AMPA, and this persists much longer. And we could show with our modeling, with a model that considers a kinetic sorption process for the AMPA, that the sorption is limiting the bioaccessibility and explains why the AMPA persists much longer than the glyphosate in soil, because for glyphosate we found a much lower sorption, so that the bioaccessibility is higher than for AMPA. And you can also use this model to look into the details on the fate of the glyphosate carbon that enters soil. And I will not go into detail because it would too long, but you can really use then the model to quantify what the relative proportion of the different where the glyphosate carbon ends up at the end of the experiment. Okay, we also did some work that's again a 1D spatial, or no, that's a 2D spatially explicit model where we look at the spatial control of pesticide degrader distribution on the pesticide degradation in the soil profile. And here I just show a short video. So we considered here two cases, or we considered here a heterogeneous distribution of microbial degraders, which you can see here on the left. And what you can see in the video is that at the places where we have a low degrader abundance, more pesticides remain. And for two different irrigation scenarios, you can see that this heterogeneous distribution, or the model can show that we can have a higher leaching risk due to this heterogeneous distribution. And in that work, we also used an upscaling approach to come to a simplified representation of the pesticide to describe the effective pesticide degradation if we have heterogeneous distribution of degraders. And you can see that the average reaction rate or pesticide degradation rate is strongly dependent on the covariance, on the spatial covariance of the pesticide and the degraders, and we can find a formulation where we describe the average reaction rate then as a function of variance covariance terms and a so-called mean field approximation. Okay, and with that, I come to some outlook, what we are currently working on. So at the moment, we are working on a soil organic matter model that considers thermodynamic controls on the use of organic carbon substances within the priority program soil systems. And I cannot explain the details here. And another work that we just recently started is a PhD project of Paul Collard, who will try to bring together the mechanistic and the data-driven modeling approaches to a hybrid soil microbial modeling approach in an attempt to make better use of these molecular biology data. To come to an end, to come at the end to much better predictions of our soil models. And another project that we are also involved in recently is focuses on the rhizosphere. And here we have meanwhile coupled microbial explicit model with a plant scale model together with a group of Andreas Schnepf. Again, I can recommend the PILS lecture here of Andreas Schnepf. And we are now able to simulate the two microbial populations around in the root system. So and this project is going on in the second phase from October on, and we will add nitrogen cycling to this carbon cycling model. And with that, I come to an end. And to summarize, I gave you a glimpse into how soil systems modeling works, and I hope I could convince you that process-based models provide forecasts of soil processes, and I showed you how they do it. And I hope I could show you that the application of models for data interpretation can increase our system understanding.